Welcome to Chapter 7. We're going to look at anatomy and morphology of crop plants. So we're going to look at a lot of different things within this chapter, specifically these four aspects of morphology and anatomy. We're going to look at the difference between monocot and dicots and what those entail as far as crop plants. We're also going to look at the plant tissue as well as plant organs. So the differences between monocot plant tissue and dicot plant tissue, same with organs. And then we're going to look at also how that affects their growth habit. When we're trying to decipher or classify different plants, specifically crop plants. So that's the justification that we will see as far as visual aspects of the plant, how that plant transports nutrients, how that plant transports different types of things to make it grow. And then another thing that we're going to look at is plant tissue and how many there are. So there are four main ones that we're going to look at, and we'll talk about those. And then we'll also look at the four organs that plants have as well, and what the difference is between seeds and fruits. So, very important when you look at monocots versus dicots. That is the first thing that plant and soil scientists really look at on an agronomy aspect. So, a monocot versus a dicot, those are two very important parts of that crop plant. So you're probably asking yourself, well, what is a cotyledon? If there's a monocot and a dicot, a cotyledon is just that seed leaf in that embryo of an angiosperm that holds the food for that developing plant. So, and then that may also then form a leaf after that germination. So a monocot has one of those cotyledons that we just talked about, and then a dicot has two. So mono has one, and then di has two. And they also share a lot of the same plant tissues as well. And so they might share those same plant tissues, but then their anatomy and their morphology will be a little bit different. And we'll look at the differences. We'll compare and contrast those differences in just a second. Work through this with you. So monocots, again, having one of those embryos of an angiosperm, and then dicot having two of those cotyledons. So monocots, let's just talk about those for a second. They're usually herbaceous. Okay, so dicots can be herbaceous, or they can be woody plants as well. Monocots have linear leaves or parallel veins with parallel veins. Okay, so their leaves will be growing up. It's going to be in a linear format. But dicots could be netted, or they will be netted, and for their ventilation as well. Monocots will have a vascular bundle scattered within their stem, but dicots will have a ring, and I'll show you a picture of those, the difference between those in just a second. Monocots will have a fibrous root system. Dicots will have a taproot system, and I'll show you pictures of those as well. Monocots will have a hypogeal germination, and then dicots most of the time will have an epigeal germination. We'll look at those in just a second as well. So what are some characteristics, or what are some different crop plants that we look at? For examples, monocots, very common monocots that we have are corn, wheat, and rice. And then dicots, we look at alfalfa. If you're looking at if you're looking at that type of the ag industry. Cotton, most common we see is soybeans. Plant tissues that we'll talk about. We'll look at the epidermis, we'll look at that vascular tissue, we'll look at the ground tissue, and then the meristems as well. So this is a diagram of a dicot plant, and we're going to kind of break those down as we go throughout the tissues as well. So, epidermis. Plants have an epidermis just like you do, and the whole point of the epidermis is just to protect that cell layer of their stems and their leaves. And one of the big issues with epidermises is to make sure that those stomatas, or those small openings, will enable that leaf and stem to aid or exchange water vapor or carbon dioxide or oxygen. So it's enabling that plant to breathe a little bit better, and that's really important in terms of crop production and how in very very hot weather how that stomata will react to that hot weather. Same with if you flip that, how that will react to very very wet weather as well. So just exchanging that water, the oxygen and the carbon dioxide, is very important when you look at the epidermis. So making sure that that stomata and those those small openings are doing the job of the epidermis. Transports that water and all of the minerals from the root system. So that plant is sucking it up from the root system to other parts of that plant. So it's a transport way to move water and then move minerals. Phylum is a little bit different because it's going to transport sucrose or the sugars and other organic compounds from one part of the plant, from the root system, to other parts of the plants. But here's the thing. When you look at dicots, because they have a vascular bundle that we talked about that's arranged in a ring, that is very important in how those minerals, the water or the sucrose, will be transported, because it's in a ring and it's not scattered. So that will happen in different forms, or that will happen in different forms and then at different times as well. So here's that description of the dicot versus a monocot. So you can see that dicot cross section. We have a very distinctive, way of looking at those vascular bundles, and then a monocot, it's kind of, it has a little bit of a pattern, but not as much as that dicot does as well. This is in your book, but I just wanted to show you what that looks like in comparison to each other. Plant. So this is the part of the plant that is doing a lot of that work, and it consists of 3 of those different types of cells: the perichlamylia, colmenclia, and the circumclia. Those 3 are very important in how we decipher what type of work they are doing. Okay, so the perichlamylia, that is found in all of the plant organs. That is very very important when we look at chlorophyll, because that's how it contains the chlorophyll within those leaves. And then energy is a very big resource in those roots and those stems. So perichlamylia is where all of that is happening. That's the job of that ground tissue. That's the job of the bulk of that plant. And then the other 2 are very important because they have those thicker wall cells, and it's going to provide a lot of that structural support. So you could imagine if you're looking at like a corn plant, those 2 cells are very vital to the structure and the way that plant is going to grow, especially in corn, because it's not just growing a very small distance from the ground. It's growing very very tall. So those 2 other cells are other cells are extremely vital to the support of that plant. Really start. So there's different types of those meristems, and we'll look at all 4 of these in class, and I'll show you different examples of them. But when you look at the apicable, that is really the growth of the tip of the shoot or the root, and I'll show you different examples of those. When you look at grasses, the intercalary is very responsible for the stem internodes and then how those leaves are then growing. Auxiliary, those are very important for the buds and the branches, and then if it doesn't have branches, then those flowers, and then the secondary, just the lateral growth that happens as well. So here's an example of a tomato plant. I like to use this graphic because it shows you all 4 of those different parts. So you can see the apicable, you can see the vascular cambium, and I'll show you, we'll do a little activity this week in those, and then the root apicable and how that is then working to be a part of that plant as well. Utilize different forms of plant hormones. So it's just that chemical that plant will produce, that will help it grow and obviously help it develop. It can be, although produced within the meristems, and that's where a lot of that science takes place. And then after that is produced in the meristems, and then it will transport to other plants. A lot of those hormones that you see there will, they're huge, they're major, and I'm going to show you some really cool science experiments that have to do with all of those and how they will grow within the plant as well.